Good afternoon, our honorable scientists, forum participants, ladies and gentlemen. Welcome to the breakout session of Life Science and Medicine. I am Cindy Chen, your MC of this session. On behalf of HKLF, thank you for joining us. We are extremely honored to have two well-renowned scientists, Professor Kazutoshi Mori and Professor Peter Walter. They will be sharing their groundbreaking research with us. After each of their presentation, there will be a Q&A session. To host today's seminar, in particular the Q&A session, we are very delighted to have Professor Alice Wong, Associate Vice President Research, Interim Director and Chair Professor, School of Biological Sciences, the University of Hong Kong, to be our moderator. Without further ado, everyone, please join me in welcoming Professor Wong to say a few words to introduce today's first speaker. Professor Wong, please.
Good afternoon. I'm very honored to be here today to moderate and chair this session. We have two very elite laureates today. Two of them, they are the Shaw Prize laureates, and with a discovery on the unfolded protein response of endoplasmic reticulum. And I'm sure with a lot of the young students here, you would know the importance of it. It is a cell signaling pathway that controls organelle homeostasis and also the quality of protein exports in eukaryotic cells. So our first speaker is Professor Mori. I will not need to read the long list of his awards, but I just want to mention that he's currently a distinguished professor and Deputy Director General of Kyoto University, the Institute of Advanced Science. So his topic today is on degradation pathway of protein misfolded in the endoplasmic reticulum. So without further ado, let's give a round of applause to Professor Mori and welcome you on the stage. Thank you.
Thank you very much for introduction. Let's start. When protein is synthesized according to the central dogma, it's a linear polypeptide. But this peptide must be folded and assembled to fulfill its function assigned by genetic code. For example, growth hormone receptor must have this beautiful structure to bind to growth hormone. Our field has one more dogma called Unfinzen's dogma. Dr. Unfinzen did a very simple in vitro experiment, and he found that this protein spontaneously folded according to the amino acid sequence. No energy is required. And then he got a Nobel Prize in chemistry in 1972. So at that time, protein folding seemed quite easy. If amino acid sequence is described in the DNA, this information transcribed to RNA and translated to the protein, then spontaneously folded to the final three-dimensional structure. But it's Unfinzen's dogma was done in an in vitro experiment. Then scientists started to show Unfinzen's dogma is okay in the cell, but it turned out that inside the cell, this protein is very, very highly concentrated, called molecular crowding. So spontaneous folding in a crowding state is very difficult. So no moving. So protein folding rely on the molecular chaperone, the special type of proteins. So secretory protein like a growth hormone and transmembrane protein like its receptor is synthesized in the ER membrane-bound ribosomes and then go inside the ER. ER has numbers of molecular chaperones quite abundantly that assist protein folding, and only folded molecules are allowed to move along to the circulatory pathways. This is a typical chaperone working in the ER. It's a heat shock protein 70 family member. It has two domains. One is nucleotide binding domain, and the other is substrate binding domain. When ATP is in nucleotide binding domain, its substrate binding domain is open. That binds to the unfolded hydrophobic patch. Then co-chaperone comes into this system that stimulates hydrolysis of ATP. ATP was converted to ADP. The ADP form, substrate binding domain is closed. By this mechanism, chaperone binds substrate quite tightly that do not allow inappropriate interaction with other proteins. Then nucleotide exchange factor comes in. They change ADP to ATP. Again, substrate binding domains become open. That substrate is released. Then released substrate are folded spontaneously according to the Unfinzen dogma. But still, some hydrophobic state stretches outside the molecule. This binding and release cycle is repeated. So chaperone grabs the substrate and release substrate, and then spontaneous folding to the according to the Unfinzen dogma. This is how chaperone works in the ER. Usually, molecular chaperone helps productive folding, but still, some proteins are still unfolded, misfolded, even after assistance of molecular chaperones. This is provided to the cell, and these misfolded proteins are retrotransferred back to the cytosol to be ubiquitinated and degraded by the proteasome. This system called ER-associated degradation. So quality control system operates in the ER. One is chaperone-mediated productive folding, and the other is ER-associated degradation. My major work is unfolded protein response. It's a cellular response to the accumulation of unfolded proteins, but today I'd like to focus on this ERAD system. Both non-glycoprotein and glycoproteins are folded in the ER by assisting of H70 type molecular chaperone BIP. But in addition, here is a glycoprotein specific chaperone system. All glycoprotein receive this carbohydrate structure: 2 N-acetylglucosamine, 9 mannose, and 3 glucose. Then this structure was attached to the newly synthesized glycoproteins. Then this glucose, 3 glucose, was trimmed to become 1 glucose. Special chaperone, glycoprotein specific chaperone called carnexin or reticulum binds to this form of glycan. Then this chaperone is connected, complexed with PDI-like isomerase, this subludo bond isomerase. Then this facilitates the folding of glycoproteins. Then this final glucose is trimmed there to make 9 structure. This protein portion of this substrate is completely folded. This was allowed to move to the next compartment, Golgi apparatus. But still, if this protein part is not completely folded, special enzyme called UGGT1 add glucose again to make G1-9 structures. Then carnexin recognizes this structure and assist productive folding. This system is called carnexin cycle. This cycle continues until the complete folding. However, still there are problems. Protein is not completely folded. degradation will start. We call the timer model. Certain time protein folding were allowed, but then system switched from folding to degradation. Key is mannose streaming. This called A chain, called A chain, B chain, C chain. Outermost mannose of B chain is trimmed, making M8B structures. Then one more good mannose is removed by next mannosides to create M7A. M7A, here is a alpha 1,6 bonded. The other is 1,2 bonded. So this new mannose structure is exposed to the surface of this molecule. The specific type of lectin molecule recognize this 1,6 bonded mannose, then targeted to the proteasome mediated degradation. So here are the two mannosides works in this degradation system, from M9 to M8B and M8B to M7A. In yeast, these mannosides are completely identified as first one was MNS1, second is HTM1. Because genetic analysis, in yeast, genetic analysis is quite easy. The knockout of NS1 causes accumulation of M9. Then knockout of HTM1 causes accumulation of M8 structure. Also, here's a biochemical analysis. MNS1 purified MNS1 convert M9 to M8, and purified HTM1 convert M8 to M7A. So completely enzymes are identified. However, how about the mammals? Mammal has one MNS1 homolog called ER1, but mammal has 3 HTM1 homologs called EDM1, 2, 3. So here is a redundant expression of mannosides. 20 years ago, to obtain knockout cells, you have to make knockout mice, then isolate knockout cells. But if you have 3 homologs, it's making triple knockout is quite difficult, laborious, and needs a lot of money. So people just skipped the knockout system and did overexpression system. Then here's ER1 works here, like MNS1, and EDM1 and EDM3 works here. But one paper showed that EDM2 has no mannose, purified EDM2 has no mannosides activity. So people forget about EDM2. We are latecomers in this field. So to start working on the ERAD, we introduced new strategies, 2 new strategies. One is knockout. When we identified 86 as a UPR sensor, then we were proposing that 86 important for the induction of chaperones. But Harvard group did a knockdown experiment. They show that knockdown its alpha beta has almost no effect on the chaperone induction. They said it is not important for a UPR system. So this was quite shocking data to us, and fields are confused. But I thought that this is a knockdown, negative result of knockdown. So we kept working on the knockout, making knockout mice and isolated knockout cells. In Y-type cells, chaperone is nicely induced by ER source inducers, but it's alpha knockout cells. This induction is quite largely compromised. Also, 86 is a major regulator of ER chaperones, showed in 2007. So we decided to knock out ER1, EDM1, EDM2, EDM3. Fortunately, about 10 years ago, we had genome editing technology, quite innovative technology came in. So we could construct a knockout cell, even normal culture cells. We use HCT116 cell. It's a cancer cell, but it's a nice diploid. So you can easily knock out 2 genes by genome editing. So another new strategy is substrate. Because this degradation Misfolded protein, people use severely Misfolded protein, like NHK, null Hong Kong type alpha-1 antitrypsin. It has a truncation in CETAM region that makes this protein quite misfolded. But when we isolate 826 as a UPL sensor, we found that 826 is also unstable proteins, half-life 2 hours. So this is a functional protein, but somehow it's unstable and constitutively subject to the ERAD system. We don't know the reason, but it has some unfolded part in the luminal region that might be recognized by this system. Then this is a degradation curve. In Y-type cell, the 86 is degraded with a half-life of 2 hours. But in the addition of kifnesin, inhibitor of monosidase, so without monostreaming, it is not recognized by ERAD system and become quite stable. So here's a huge difference. So analysis will become easy. So we created even 1, 2, 3 knockout cells and analyzed carbohydrate structure. This is quite complicated, so I show that only difference between Y-type cells and knockout cells. When Y-type cells are treated with kifnesin monosidase inhibitor, MAN9 structures accumulated. Then M8B to M7 conversion was thought to be done EDEM1 and 3. The knockout of EDEM1 caused small amount of small accumulation of M8 structure. Most likely, knockout EDEM3 caused large amount of accumulation of M8B structure. So usually before that, EDEM1 was a major player, but the knockout cells show that EDEM3 is major player in this conversion from M8B to M7. Then ER1 was thought to play in this conversion from M9 to M8B, but we found that it's only minor effect. Indeed, ER1 is not local in the ER. It's local in the Golgi. So they cannot participate in this ERAD system. Somehow overexpression causes artificial result. Then we found in EDEM2 knockout cells, M9 structures accumulate like similar to monosidase inhibitor treated cells. So, and this degradation of 826 was attenuated the EDEM2 knockout cells, then 3 and 1 in the order. So our genetic analysis showed EDEM2. Previously, it has shown that it has no monocyte activity. Nobody worked on this protein, but our genetics showed EDEM2 is a key player in the ERAD system by initiating MAN9 to MAN8B conversion. Then EDEM3 and EDEM1 is a major player in this second monostreaming steps. But of course, you also that here, why EM can play in a major role without monosidase activity? We know that, and our competitors claimed, even after publication of our paper, this group showed that EDEM2 has no monosidase activity. How overcome with this conflict? Then, hint as in the E system. I skipped the details. HTM1 alone has no monocyte activity. But if HTM1 is complex with PDI, it shows enzymatic activity. So we thought that EDM2 must make a complex with some other protein to get enzymatic activity. EDM2 has this system. Each HTM1 has a disulfide bond in the monocyte activity domain. Our EDM2 has many cysteine residues, and we make a mutation, converted cysteine to alanine. Then all cysteine was converted to alanine, and found that when these three cysteine was mutated to alanine, degradation activity was lost. This C65 and C408 exist in the monocyte homology domain. That makes the disulfide bond. Then this C558 must be complex to be PDI. The problem is ER has a dozen of PDI-like family members. We thought that we have to make all of the knockout. But fortunately, 2016, this paper came out. This group did a genome-wide screening for looking for the EDAD component, and found that here is a EDM2. And in addition to PDI, here is a TXNDC11. It's a cytotoxin family member, contains CXXC catalytic motif. So when SDS gel was run under non-reducing condition, EDM2 exists as this one, monomer band, but in addition, he has a high molecular weight band. This EDM2 must be complex with some other protein via disulfide bond. So we checked PDI, anti-PDI antibody did not react with this complex, but anti-TXNDC11 antibody reacted nicely to this upper band. The immunoprecipitation showed that TXNDC is immunoprecipitate with Y-type protein, these two cysteine mutant, but did not immunoprecipitate with this C558 proteins mutant. Now this cysteine is complex with TXNDC11. We constructed TXNDC knockout cells, and in this high molecular weight band was disappeared. Also, degradation of this ERAD substrate is delayed in the absence of TXDC11. So TXDC11 is the actual part of this ERAD system. This TXDC11 has many cysteines, more than 20 cysteines. My graduate student, Jinto Georgi, mutated all of them, and then found that one here, in this mutant, C692 mutant has no complex. So we found that C558 of EDM2 is disulfide bonded to C692 of TXNDC11. So we try to purify this complex and try to show the enzymatic activity. When we did a Western blot, TXNDC11 is always appears a doublet band. We found that here's the two methionine. Both around this methionine is meet Kozak sequences. So we saw that this protein translation may start from first methionine or may start from methionine 58. Then if methionine 58 was mutated to alanine, or translate start from first methionine and show the upper bound. Then if when first methionine was mutated to alanine, this translation starts from methionine 55 and shows lower bound. Okay? Then if this TXDC was translated from first methionine, this hydrophobic stretch will function as a transmembrane domain. So almost all protein is recovered in a pellet fraction. But if TXDC was translated from methionine 58, this this hydrophobic stretch may or may not function as transmembrane domain. So now half of the protein is recovered in the supernatant. So it's easy for the biochemical analysis. So we purified this complex and compared Y-type mutant, which make complex with TXNDC, and C558 mutant, which cannot make complex with TXNDC11. Then here is a substrate, MAN9 oligosaccharide was incubated wild-type ADM2. This MAN9 was converted to M8. Then after 12 hours, almost all M9 was converted to the M8 structure. So this is much more active than East HTM1 pin. Just clear ADM2 complex has a clearly monocytase activity. So uncomplex ADM2 has no enzymatic activity as previously shown in 2005. So this ADM2 alone has no monocytase activity, but ADM2 TXDC complex has a monocytase activity. So this is show that this M8 is M8B. So now TXDC11 and EDM2 is a fast monocytase in the ERAD system. We also checked ADM3 and ADM1. They are not complex with TXDC11. So we purified EDM1 and EDM3 and did a biochemical reaction. The purified EDM3 convert M8 to 765. It's highly active, much more active than EDM1. So this is consistent with genetic analysis. So we identified enzymes responsible for the first mannose trimming step and second mannose trimming and pathway to the degradation. Finally, I little bit talk about UGG1 system. As I mentioned, that UGGT1 de-add glucose to when this protein part is not completely folded. But previously, Dan Hubbard showed that this deglucosylation is important to folding, but not important to degradation. That was proposed by Dan Hubbard. So we confound his result. Here's a deoxynitrogen, the inhibitor of glucose trimming that do not affect degradation. But again, we did a knockout experiment instead of the inhibitor experiment. Here is UGT has two homologs, UGT1 and 2. We construct UGT1 knockout, 2 knockout, and double knockout. Then checked degradation with ATF6. We found that ATF degradation was accelerated UGT1 knockouts or double knockouts. UGT2 knockout has no effect. The UGT1 accelerate degradation of ATO6. That means carnexin cycle and ERAD is competing. So also this is another typical ERAD subset, NHK. NHK degradation was accelerated by UGT1 knockout. NHK-QQQ is a non-glycosylated mutant of NHK. No glycosylation occurs in NHK. In that case, degradation was not affected by UGT1 knockout. The NHK degradation is delayed by overexpression of carnexin cycle proteins, carnexin and UGT1. On the other hand, NHK degradation was accelerated by overexpression of ERAD component, EDM2 and EDM3. But this accelerated degradation was delayed by overexpressing of carnexin system. So here's a tug of war between carnexin cycle and ERAD. So then question is why deoxynitrogen inhibitor did not affect degradation. So in the inhibitor, deoxynitrogen is an inhibitor of glucose trimming. So when white type cell treated deoxynitrogen, this glucose-containing oligosaccharides increase. Then MAN9 and MAN8 structures decrease as expected. So this glucose trimming is blocked by deoxynitrogen. But we found that, here's a decrease and here's an increase. Then we found the interesting increase in GM8 structures. So mannose trimming occurs even in the presence of glucose. So deoxynitrogen inhibits glucose trimming, but cannot inhibit mannose trimming. So this G1M9 was converted to GM1 by EDM2. Then probably converted to the GM7 structure by EDM3 and EDM1. Then this structure recognized by specific lectin and subject to the ERAD. So this last slide. So when newly synthesized protein is folding is assisted by first by BIP, HSP70 type chaperones. Then glycoprotein enters carnexin cycle to assist productive folding, and folded molecules are moved along to the secretory pathway. But this carnexin cycle is not effective for the folding some circumstances. ERAD compete with carnexin cycle and extract substrate from carnexin cycle to degrade to endure protein quality control system. That's my story. Thank you very much.
Thank you very much. Very insightful findings. Maybe as a chair, I got the liberty to ask the first question. So I just wonder, you mentioned the importance of the glucose as well as the mannose in the glucose and the mannose in the glycoprotein, right, in the folding of it. So how about in the context of chronic stress or like stress and aging? How does the yell's capacity to manage these unfolded glycoprotein change over time?
Relationship between aging and ERAD is not known properly, but UPR is very important for aging. And the UPR has 3 sensors. IRE1 and PERK and ATO6. The IRE1 is conserved from all eukaryotic cells, from yeast to human. Knockout of IRE1 in yeast and C. elegans and Drosophila melanogaster shorten the lifespan. So this maintenance of UPR is important for the preventing aging, I think.
I see. So it's evolutionarily conserved. Okay, good. Questions from the audience? Yes, please.
Thank you for your talk. So just a very quick question about the pathway itself. So you mentioned that there's a kinesin cycle for unfolded proteins, but when it becomes folded, like how do it exit the whole cycle from the kinesin cycle?
Can I go back? Yeah, somehow this is a gatekeeper. UGT1 is gatekeeper. This enzyme somehow recognizes unfolded state and add glucose. But if protein portion is completely folded, UGT1 cannot do anything on this substrate, and this go to this protein go to the COP2 vesicle and then transport it to the Golgi. It's not known. Yeah, it's Arrhenius worked very hard on this protein, and somehow he could show that this protein recognizes unfolded state. But we want to work on how EDM2 works on the EDM2 can discriminate for the protein or unfolded protein. But it's very difficult experiment because we have to purify substrate with only M9 structure. They get denatured. But the purification protein with only M9 structure is difficult. Technically difficult, so it's a good question. But we want to know your answer, but we don't know yet. Thank you.
Yes, Prof.
Thank you. So you mentioned about the competition between the kinesin cycle and also the URAT. So in physiologic context, how this competition works, like the UGGT protein will have degraded, or how the competition really works in cells?
Just a timer model. So this folding cycle if folding takes time, the substrate stays in a year or longer period. Then the chance of the EDM2 to meet substrate increase. Just a competition. So if substrate stay longer, more chance for EDM2 react on the substrate. Just extracting substrate from calnexin cycle. That competition we mean.
Very nice presentation. Thank you very much. So you give a very beautiful story in terms of how EDAM is involved in the clearance of glycosylated protein. So we know that EDAM is activated through the unfolded protein response, right? And there would be instances where protein is folded, not glycosylated. So under those sort of scenario, how does the system clear unglycosylated proteins?
So this EDM system is a target of UPR, and when ERCs occur, misfolded protein accumulate more than normal condition, UPR is activated, induce EDM. First, also, BIP is a major target of UPR. So both chaperones and ER components are increased. So I think competition occurs. And then, but anyway, cell try to lower the amount of misfolded protein in the ER by means of chaperone mediated assisted folding and also the ER system. Yeah, no matter what conditions, cell won't decrease the amount of misfolded proteins.
Hi, I'm Zichen, and thank you for the nice talk. I'm just curious about why you chose about his shock protein 70, because his shock protein contains small his shock protein 60, or his shock protein 90. Why you choose this one? I just curiosity.
No, I didn't choose.
Oh, okay.
So this chaperone was originally identified as heat shock inducible protein. If you add heat shock, like increase temperature, culture temperature from 37 to 42 in E. coli and human, or 30 degree to 37 degree in yeast system, that called heat shock. That under heat shock condition, several proteins are transcriptionally induced. That's why it's called heat shock protein induced by heat shock. Then here's in the cytosol, HSP 90 type, molecular weight 90 kilo-K, 70 type, 60 type, and then 40, and then small heat shock protein. ER has HSP 70 and 90 only. And then this HSP 70 is a major chaperone. This is essential protein from yeast to human. HSP 90 is not essential protein. It has a special client protein. But this HSP 70 works for everything. The first interactor with newly synthesized protein entering in the ER. So that's the system. Okay.
Further questions? Yes, please.
Hi, Professor. Very nice talk. I'm very curious about if I overexpress extracellular proteins, but I remove the signal peptides. They are glycoproteins and overexpressed, and I found they can really overexpressed. What do you think? Can they fold correctly?
Yeah. Overexpression of normal extracellular matrix protein induce UPR. So try to increase the amount of chaperones, then to try to defold it. I identified ATX as a UPR sensor. Then its knockout cause embryonic lethality in the mice. Mice without ATX died very early. So we could not find any ATX knockout in even E8.5. So we could not find any reason for the death. Then we changed the system to fish lab, Medaka fish. Then we found that 806 knockout caused block development of notochord that synthesize large amount of extracellular matrix protein. To synthesize large amount of extracellular matrix protein, they need notochord need ATIS-mediated induction of chaperones. So if chaperones sufficiently induced, your problem cleared. But if chaperone is not enough, so overexpression external matrix protein may cause UPR and then may cause cell death.
Just one extra question, so maybe a bit stupid question. So, can the ubiquitin ligase target or ubiquitinate the protein in the M9 stage?
Ubiquitin ligase exists in the ER membrane.
In the ER membrane, right? But in the M9 stage, can it target in the M9 stage, like with in the calnescent cycle, can the ubiquitin ligase still target the protein there?
But ubiquitin E3 exists in the ER membrane and active site in the cytosolic side. So it cannot work on the luminal side. Just the cells must first translocate to the cytosol, then ubiquitinated and degraded by proteasome, maybe directly by proteasome talked about in the morning session, but we don't know. So detroscation first, then ubiquitinated.
Okay. Yeah, last question. Last two questions, okay. last year.
Enough
So
time.
you mentioned about the Arvinson hypothesis at the beginning, mentioning that no one did the folding experiment in the presence of the crowded environment. So are you aware of any experiment that to try to look at the protein folding in the crowded environment? The protein folding in a crowded environment. So has anyone tried the Arvinson hypothesis in a crowded agent, something like that?
Yeah, the crowded proton folding is very difficult. It was shown. And I think dogma is right, because now computer predicted the final structure based on amino sequences. So the AlphaFold is the AI-based new technology, but then predict precisely the final structure based on the amino acid sequence. I think then dogma is right, but it's difficult to occur inside crowded cells.
Okay. We are given the time. Yeah, maybe. I'll get, if we have some time behind, we can have another round of questions. So thank you again, Professor Mori, for your very great talk with us. Yeah. So our next speaker is also a elite scholar, Professor Walter, on the unfolded protein response, and he's distinguished professor in the Department of Biochemistry and Physics at UCSF. He's the founding institute director and still currently the institute director for the Altos Lab in the Bay Area Institute of Science, and he's also a Shaw Prize laureate. So today, Professor Walter will share with us his work on targeting the cell's stress pathways for therapeutic benefits. Let's welcome Professor Walter.
Well, first of all, I would like to thank the organizers and the Li Shaoqi Foundation and the Shaw Prize Committee for making this all possible, right? I mean, we've been having a wonderful time here, and I think a big applause for everybody here who contributed making this such a wonderful event. So what I want to do is expand basically on what Kazu told us about, but bring it a little bit more towards the human application. Because these pathways are so important for our health. As you already heard, unfolded protein response breaks down as we get older. And as we get older, I may be the oldest person in the audience here, I think we can agree that the biggest fear we all have, that we lose the ability of being human by losing our memories. So part of learning and part of our brain function is incredibly important for us to be who we are and to enjoy life and to enjoy life with each other. So I want to start this talk with a dream, and I hope you can all join in that dream, because it is one that is not unreasonable to have. What if we could figure out what defines a young, healthy cell? And if we could take that information and devise ways to maintain cell health longer, so that every one of the 10 to the 13 cells that we have in our bodies basically learns and maintains the ability that we have in a newborn baby to heal and to be resilient to change, right? The baby scratches itself, and the wound heals, and there's no scar tissue. It is all seemingly perfect. So if we can maintain that state longer, then we can use this knowledge to approach biomedical research in disease-agnostic ways, based on that cell biology, and nudge our current system, which we really don't have a healthcare system, right? We have sick care. We go to the doctor when we are sick, into one of actual healthcare, of being proactive. Of course, we still want to deal with disease, and the ideal way here is depicted in this wonderful painting from Lucas Cranach of the Fountain of Youth, where you see the old people here entering this pool of some magical substances and emerge on the other side, resilient, rejuvenated, and very attractive. So where can we do this? I mean, both Kazu and I embarked on this journey through the unfolded protein response, and other stress pathways, I think, confirm what we have learned here, or some of them actually have been ahead of us, in that these stress pathways are really marvelous in that they regulate cell health by controlling the homeostasis of the various components. And they make sure that the system is in proper balance. They sense deviations from homeostasis. And that's the important point here, right? When something goes wrong and unfolded proteins accumulate, these pathways are activated, and therefore they mark sick cells, right? They mark cells that are no longer doing their job properly. And we can, for discussion's sake, we would like to define this balance as the useful state. And we can come back to that in the discussion. So they mark sick cells, and because they mark sick cells, and they're actually not that important in healthy cells, we can inhibit them, we can manipulate them with very little toxicity. And this turns out to be true over and over in different examples that we've looked at. They make life-death decisions. Kazu pointed to that the unfolded protein response is at the center of deciding whether a cell can fix the problem or should it kill itself, which is, of course, a fascinating question biologically of how can a cell make that decision? It's binary, right? And that puts them in the center of many different human diseases, and many of these are age-related diseases. And the list of these disease classes, is really numerous and very long. We have cancer. Cancer cells evolve in a part of your body, become selfish. They become genomically unstable. They make a lot of mutations and protein complexes. Chromosomal imbalances cause protein complexes to be assembled in the wrong proportions. All of these deviations are being seen by the stress pathways, lead to activation. And in most cases, this leads to cell death. But in cancer, the cell death does not occur. This decision is made wrongly, and now the stress pathway comes in, such as the unfolded protein response, such as the heat shock response, and buffer the cell and help it survive when it really should die. And this is a property of every cancer, right? This is not cancer specific now, so we can broadly apply that to a whole class of diseases. Cognitive disorders, that's where I'm going to focus my talk as I go on. Neurodegeneration, diabetes, metabolic disorders, inflammation, and we could extend that list endlessly into many, many different disease classes. So we have an opportunity here by learning about molecular events in the cell of affecting medicine in a very, very broad spectrum of ways. So I'm going to focus on cognitive disorders and how that comes in and links to protein quality control in the cell is based on a discovery that was initially made by Mauro Costa-Matthioli when he was a postdoctoral fellow in Nahum Sonnenberg's lab at McGill. And what Mauro discovered is that for all of us to form new memories and to store them into long-term memory, we need ongoing protein synthesis. This was known. Goes back all the way to Eric Kandel and learning in aplesia and goldfish and mice. But what Mauro discovered is that this step of protein synthesis can be controlled by a phosphorylation event of this very central translation initiation factor in the cell, ERF2. And ERF2 can become phosphorylated and then basically diminish the translation, which diminishes the ability of an organism to form new memories. And this is controlled. One of the kinases that mediates this phosphorylation event is PERK, one of the three pathways of the unfolded protein response. I'm going to get back to that. Now, the way this discovery was made was genetic. So what Mori did is he made a heterozygous mouse in which one of the alleles that becomes phosphorylated here is changed from a serine to an alanine. So this mouse can only make half as much of the phosphorylated form of this inhibitor, of this bad form of a translation factor. And the surprising result was that these mice learn better. That's an amazing result, right? Here you have a mouse, you make a mutant in it, and you actually improve upon the wild type, upon the behavior that evolution doesn't seem to have optimized, or at least there is a way of going beyond that. Now, we happen to have in the lab, and this pathway, this branch, the side branch of the unfolded protein response is called the integrated stress response. We happen to have isolated in the lab inhibitors, small molecules, that block the various branches of the unfolded protein response. And for this ISR branch, we had isolated this molecule, which we call ISRIP, for integrated stress response inhibitor. And this molecule basically does the same thing. It inhibits this phosphorylation from being effective in the cell in shutting down translation. So having this molecule, we just thought, well, let's send it to Nahum's lab and see if it also helps to improve memory. And they did the experiment. The memory is measured here by this classical Morris water maze, where you put a mouse in this trough of milky water. It has to find this hidden platform and remember where to swim. It orients itself literally by posters on the wall, the visual clues around the room. And what you see in this assay here is as this mouse learns over a couple of days of training, eventually it plateaus, reaches a maximum performance. And if that mouse was injected with the ISRIP, it learns substantially better. This really, really remarkable result, I think. And this curve very much overlays with the genetic result. So for everything I'm going to tell you here, we have the pharmacology precisely paralleling the genetics, which is very reaffirming. So what this tells us is that the integrated stress response, via this phosphorylation event, inherently limits memory formation. And if we give the drug, or we make the mutation, we take this break away. And now we are free to. So ISRIP is a cognitive enhancer. What is it? How does it work? It was isolated, or the integrated stress response, as I told you, is this general translation factor here, EIF2. It's a GTPase, and like many GTPases, it can flip back and forth between a GDP and a GTP bound state. In the GTP bound state, it picks up methionine initiator tRNA and then brings the methionine to the AUG in the ribosome, pretty much every mRNA that's being translated in the cell. So it's a very generic event. But like most GTPases, this exchange isn't spontaneous, but it's catalyzed by an exchange factor. That exchange factor, in this case, is this huge complex of 10 subunits, 5 different kinds, 2 copies each, so it's two-fold symmetric complex, which catalyzes this reaction. Now, what phosphorylation on this single serine residue of the alpha subunit does is it renders the substrate of the exchange factor into an inhibitor. It now binds to the exchange factor, and by binding to the exchange factor, it pulls it over out of this reaction. It forms an inhibitor. We have much more EIF2 in the cell than we have exchange factor, so a little bit of phosphorylation can go a long way of saturating that component and inhibiting it. And as I already told you, this phosphorylation event is mediated by kinases that respond to stress conditions in the cell. In this case, we have ER stress mediated via PERK. We have a double-stranded RNA accumulation reflective of viral viral infection of cells, nutrient sensing via GCN2, and redox state or mitochondrial health being sensed by the HRR kinases. And all of these kinases converge on that single serine residue. That's why it's called the integrated stress response. Integrating between all of these deviations from homeostasis and eliciting a common response, slowing down translation. Now we screened for this molecule, ISRIP, and ISRIP basically makes cells immune to this phosphorylation event. So they can keep on translating, so it blocks the integrated stress response. This is what it looks like. I already showed you the structure. It was isolated by Kamala Sadrowski when she was in the lab. She's really a remarkable scientist. And I think it's a very beautiful molecule, right? It's two-fold symmetric. Both of these arms are identical in their chemical nature. They substitute this cyclohexane in the middle here, which can be substituted in cis and in trans. The cis form has no activity whatsoever. The trans has all the activity. So we have virtually identical chemical matter that can be used as a control in our experiments. We solved the structure of ISRIP bound to our exchange factor. This was in collaboration with Adam Frost and his lab. And here's a structure that we obtained a number of years ago by cryo-EM. And it's a beautiful structure. And there in the middle, we have our ISRIP molecule. The blue is the data. The golden halo here is photoshopped, just to point the thing out for truth of advertisement. But based on this structure, it immediately suggests what this molecule may be doing. Right? It fits beautifully into this deep pocket, and it links this symmetrical complex, staples it together from different portions, from different subunits. So it's a central portion there that prevents this complex from opening up this interface here, which we now know is part of the regulation by which the exchange factor works. And what ISRIP does and what the exchange factor does is this big complex goes through a series of conformational changes. It's an allosteric protein complex that when the inhibitor is bound to one phase at the bottom, the phosphorylated form, it pulls apart this interface here where the translation factor would have to bind as a substrate to become activated by the exchange reaction. So it's this allosteric flipping back and forth, which we call our butterfly model, the butterfly there flapping its wings, that we think is the central of ISRIP action here. And we can now, with the beautiful power of cryo electron microscopy, single particle, really see the side chains at this interface, how they go through this beautiful dance of changing from one conformation to another, which now opens up many opportunities here, right, to throw monkey wrenches in this molecular machine and come up with additional molecules, small molecules, potential drugs in the future to block this process. This was done by Rosalie Lawrence, postdoc in the lab, in collaboration with Susan Marxey at Berkeley. We can extend this a little bit further. Very recent work by Advait and Udit in the lab, coming up with this whole communication axis that is down at the center of this molecule here, flipping back and forth, and showing you all this beautiful, it's an orchestra, right, of the side chains going from one state in alternative states. And what this flipping back and forth of the structure does is it flips the exchange factor from an inactive state where it can bind the phosphorylated form, or binds the phosphorylated form, into the active state over here, which is promoted by ISRIP, and which is the interface has to be tight in order for the exchange reaction to occur. So we understand the action of this drug at at atomic detail in very, very high precision at this point. So can we use this, since it is a cognitive enhancer, can we use this information to open new paths to or explore potential therapies in conditions with cognitive defects? So we collaborated with Susanna Rossi at UCSF. She was at the Trauma Center, who has this model of traumatic brain injury in how it affects learning. In this model, basically mice are subjected to a mechanical insult, a mechanical injury to the brain. And then they learn how to navigate this modified water maze, having to find the hidden platform. This mouse is a control mouse. It has learned how to find the platform. This mouse here is one that has been afflicted by this injury. It is basically swimming a random path, eventually finds the platform, but it is demented. It cannot form new memories in the training period. Here's the experiment I want to show you. We inflict the injury, then we wait 4 weeks for the mouse to recover, to subdue any inflammatory reactions. And now the mouse is fine. It looks fine. It swims at the same speed. It shows no defect, but it cannot learn anything. It is absolutely unable to form new memories. We then go through the training period here, and either injecting the drug or not. And then after 3 injections, we do a memory test. We wait a week. We do another memory test. And as I said, if we didn't do anything, these mice would stay demented and unable to learn for months to come. Here are the data. Injured mice don't learn over this first day of training. Control mice learn fine, making fewer and fewer errors as the day goes on. The next day, the mice that are injured still don't learn. The ones that are the control mice remember what they did the day before and hardly make any mistakes anymore. A day later, they remember. A week later, they remember. There's no drug here. These are just the controls. Now we add the drug to the system, and already on day 1, you see how the presence of the drug improves the memory as the mice learns where to find the platform. And then on day 2, it's a miracle, right? The mice that have been injured now become indistinguishable from the control group. A day later, they remember. A week later, they remember. And this has a high statistical significance, so it's really a very, very powerful result. I want to show you one more assay here, and that's an assay which measures anxiety or risk-taking in the mice. You put the mouse in the middle of this cross-platform here, where the mouse can choose between being in these hidden protected compartments in the dark, or has to venture out onto this little platform here, and then could potentially fall down. So the experiment that was done here is repetitive mild brain injuries. So these are injuries like a football player would experience in the field. If they're repetitive, they are additive, so it becomes really a big problem as it happens over and over again. These mice, again, become completely demented. They don't learn at all anymore. We again let them sit for 4 weeks. We then inject the drug. We subject them to this test. And then we let them sit for another 3 months. No more drug. The half-life of the drug in the mouse is 8 hours. So there's no chance, or it's very implausible, that there is a reservoir of the drug. And we do the test again. Here's what the data look like. You measure the time in the open, so exposed. You see that the injured mice are much more risk-taking than or less anxious, risk-taking than the control mice. And if they've been injected with the ISREP, that effect is completely reversed. And if we wait 3 more months, the effects of the drug, the effects of the injury remain, but the effects of the drug treatment also remain. So it is as if we had pushed the reset button, right? The system, these mice have been cured of the defect. This is not learning per se. This is not memory formation, right? Because the mouse wasn't trained. We just put it in the middle and we watch what it does. It's innate behavior. So this is a readout for brain health, basically. Things are no longer working properly. We can continue this into other models of age-related diseases. We can take old mice which have difficulties learning. We inject the drug. They learn like teenagers again. So wonderful, wonderful efficacy. Now, can we correlate that with some aspect that we can actually observe in the brains? And you know, learning is mostly about plasticity of the synaptic connections between neurons. So when one neuron, the axon, connects with dendrites in the postsynaptic neuron and then transmits the chemical information across the synaptic space. These junctions, the synapses, are formed between boutons in the axons and dendrite. The dendrites have spines that come out. So a very adventurous and very, very hardworking graduate student, Elma Frias, in Susanna Rossi's lab, decided to set up an assay where we can actually monitor the timing and the birth and the degeneration of these dendritic spines in mice. So what she did is she took mice, subjected them to the injury again, and implanted a window into the skull so we could repetitively find the same neurons, the same dendrites in the brain, and follow them over multiple days and see what happens to this dynamic of these spines that are trying to form new connections in a learning regime. And the amazing result, so here basically you can see that at different days after the injury, here or even in a normal animal, but it's post-injury here, we can distinguish different kinds of these dendritic spines, and you see we can find the same view again. We can identify the same neuron, and there are some spines that are stable because they have formed productive synapses with their corresponding axons, and then there are some that are remodeled. And this happens in a learning profile. It has to, or in a behavioral profile, has to happen dynamically. There are some spines here, like these guys, that have newly born. And then there are others, like these guys here, which in these few days have disappeared. So there's a dynamic of these things that are, as they are trying to find the partner of forming a synapse. Now we do this experiment, again injecting the injury, doing the surgery to put the window in, then have various imaging sessions over time, plus minus drug injected later on. So the first two are to get a baseline, and then we look further downstream. You see that in the presence of the brain injury, spines become much more dynamic. Many more are being born, trying to find partners in the axons. And this lasts over the whole period of the experiment here. But if we now add the ISREP, you see that these newly born, these abnormally dynamic spines that are being formed is being reduced back to normal level. So basically what happens is in the presence of the injury, the spines form, but they cannot find the proper connection. The synapses are not stable. The spines retract again. So they're out there trying to find their partners but don't succeed in doing this productively. And as we add the ISRIP to the system, it very quickly normalizes back to. This is the first evidence, right, that we have a real neuroanatomic change in the brain that we can quantify and monitor that relates to the learning experiences. So the summary, then is a brief treatment with ISRIP. Weeks after injury restores cognitive and behavioral loss. Concussive brain injury profoundly alters spine dynamics, measured up to a month after injury. And the brief pharmacological inhibition of the ISR reverses these structural changes and associated with the working memory defects that we measure in these behavioral assays. Now, this is the most important slide I'm going to show you. And basically says that in all of these different conditions that affect memory, cognition, diseases, terrible diseases, right? Alzheimer's, Parkinson's, ALS, frontal dementia, including other conditions like the mechanical injury, traumatic brain injury, Down syndrome, a condition in which we have chromosomal abnormalities, right? Extra chromosome, complexes, proteins get made in the wrong proportions. The ISR is induced. I already told you age-related cognitive decline, the mechanical injuries. Under all of these conditions, we know that the integrated stress response is induced. And under all of these conditions, we know that if we block it, we restore brain health. So we think then that it's really this stress response, not the inducing conditions per se, that is the culprit for the cognitive defects or behavioral defects that we are seeing associated with these diseases. I've also showed you that we have this phenomenon where the response feeds onto itself, and once it's on, it keeps going for months after the traumatic brain injury. But if we add some maladaptive feedback loop, but if we inhibit this maladaptive feedback loop, we reset the system to normal. So this is basically where we are. This is very good news for the mice in the audience, but mouse brains are very different from human brains. So we hope that this will translate into real medicines. The progress report is that ISRIP analogs have been produced by different companies. They've been picking up on that. We've been working with Calico and Abbey. We licensed ISRIP to them, and actually Carmela, the person in my lab who discovered ISRIP, is now in charge of the clinical development there. Denali decided to compete with Calico. They've passed phase 1, so toxicity studies are fine. There's no toxicity that has been observed. And ISRIP analogs are currently in human trials in individuals with a disease, a genetic disease called vanishing white matter disease that affects young children. And it's the lowest bar because nothing can be done for them currently to pass into the clinic and major depressive disorder, which is probably more related to the anxiety phenotype, the risk-taking phenotype. Now, I think Confucius said that there's beauty everywhere, but just most people don't see it. So we made a beautiful bronze casting out of the complex here with a little golden ISRIP in the middle. I worked with a foundry in Berkeley to realize that. Here is my friend Sebastian Bernales. Take-home messages for you. We inhibited the cell stress pathways. We think it's a clinically very promising approach, spanning a wide range of different diseases. We identified ISRIP as a first drug-like molecule that blocks the integrated stress response. And we understand its function in atomic detail, so we can easily guide in a rational way, develop further pharmacological development. We showed its efficacy as a cognitive enhancer and identified with it the integrated stress response, or the side branch of the unfolded protein response, as a druggable pathway and as a promising avenue to combat cognitive disorders, and shows this amazing efficacy in this broad spectrum of models of cognitive loss, brain health, and the dynamics of synaptic remodeling. And the important thing for all of us, for all of you, here is that we had no idea where we were going, right? We did not plan to get into cognitive dysfunction. We could have never written a grant saying that. They would have chased us out of the room. They would have laughed us out of the room. So this all evolved as a result of curiosity-driven, disease-agnostic basic research. And this is what our funding agencies need to hear over and over, our reviewers have to hear over and over. And you should think about your own careers, right? That it's the steady production of knowledge that eventually leads to discoveries. That you cannot plan for discovery. You cannot plan for for winning a Shaw Prize in the future. These are things that are the product of hard work and at some level serendipity. But serendipity is not dumb luck, right? Serendipity means you have to be prepared for the unexpected, and when the unexpected happens, take advantage of it, right? You don't let it go. Then bite in and work even harder. So one last thing, let's not leave here and say that aging is always such a bad thing. So this is my evolution from graduate school to who I am now. Thank you so much.
Thank you. Thanks a lot, Professor Walter. I think, already a lot of questions. The ISRIB inhibitor, the promising effect of it is very exciting. So can I just start asking, what are the main challenges like developing drugs like against the cellular stress pathway that you would see like without affecting normal cell behavior?
Yeah. But the main challenges are obviously now in clinical trials, right? To find real efficacy and real improvement in the conditions in a patient, so that this hurdle can be overcome. I think we've solved it molecularly. There is not much more for us to do here, but to explore potential other avenues. I mean, ISRIB was a really lucky find in that we have no side effects, right? It emerged from the screen right away as a very potent agent. It was brain penetrant. It was had a reasonable half-time in the mice, so we could actually do this experiment. And here's another lesson for all the young folks here, in that we did not know ISRIB would go to the brain when we sent the drug to Nahum Sonnenberg's lab to do the first learning experiment here. So they were just adventurous to say, okay, we'll try it. Let's see what happens. So this is risk-taking in your experimental approaches. Just go for the jugular and leave the capillaries alone when there is an opportunity to do so.
Yeah. Could we make it a personalized medicine, tailoring, like targeting certain specific cellular pathways for individual patients?
That would be fantastic if we could get there. On the other hand, if there is no negative consequence, right, we may not need different drugs for different people, which of course is a big advantage. I mean, many of us take the statins these days, right, and the positive outweighs the negative so much. I would not. I mean, the companies improved the ISRIB, right? They made it a better pharmacological agent, so you can actually have it orally delivered. And I met a little boy who has this vanishing white matter disease. He's in one of the clinical trials, and he has improved tremendously after being in a clinical trial for this. There's no readout from the trial yet, so we don't have the statistical data. But just from a personal note, it is really heartwarming and touching to see him recover.
Sure. Yeah. I'll open the floor for questions now. Maybe ladies first.
Hi, thank you so much for your wonderful talk. My name is Rafaela, and I study Parkinson's disease at the University of Toronto. I have a personal history with studying ISRIB, actually, using it to rule out EIF2 phosphorylation in the cellular pathway that I was studying 10 years ago, BDNF to ATF4 upregulation. Yep. And so I actually have like a neurodegenerative disease sort of related question on your talk. Because you looked at very short term after brain injury for the mice, like kind of like 37-day, maybe 1 month, 2 month time points, with or without the treatment of ISRIB. I was wondering if there are like, in terms of the brain injury induced like tauopathy, like, or other sorts of pathology in the long term, like within like maybe the year span of these mice, and if like ISRIB treatment could also prevent these neurodegenerative disease-like things happening in your mouse model.
Yeah. So we have not tested this in the longer time frames here, which is all what needs to be done now, right? And we actually most tests for traumatic brain injury and the resulting cognitive defects for different clinical trials were done right after injury. So we are already waiting a month here. So we know that everything else is fine or has been recovered, and we can still reverse it at this stage. But this is as far. You've basically seen what we have here, and I don't know. Calico probably has done lots more studies that are not accessible to us at this point. Regarding other cognitive disorders, the neurodegenerative diseases, once the cells are dead, right, in Alzheimer's, for example, I don't think we have a chance of recovering them. But early treatment, I think you hit it on the head here, is precisely where we want to go, right, and become preventive, ideally, right? It's just very, very hard to do in a clinical setting as tests.
The one red T-shirt.
Thank you. I have maybe 2.5 questions. First, how much this effect related to apoptosis or cell death? If so, can they like kind of a cell death inhibitor have this kind of similar effect you will expect?
But the integrated stress response triggers cell death, right? If it's on for too long, for too strong, you get, because you shut down translation.
Exactly.
It leads into
so do you expect
pathways. You induce CHOP transcription factors, ATF4, that become pro-apoptotic.
So cell death inhibitor might have the similar effect?
I don't know.
Okay. The second question is like, can you also observe a little bit more like protein aggregation or DNA damage if you treat the cells with the ISRIB?
We have not looked. I can't comment on that.
Okay.
I don't know. That was a half question?
Yeah, maybe.
Thank you, Professor Walter. I am Sheena from the Hong Kong University of Science and Technology. My first question is, I would like to know the dosage of the ISRIB placed on the mice, and also have you considered checking male and female mice, because it might affect the results for different genders, maybe due to different hormones?
That's actually a very interesting question. So so for traumatic brain injury, it makes no difference. So this is close to Susanna's heart. She always asks, have you tested in females too? But for the anxiety assay, it does make a difference. Oh, yeah. So female mice, you can, when they're subjected to the same concussion treatments, they don't care. Oh, yeah. They don't become more risk-taking or out there. So this is an effect that is sex-specific.
Yeah, and one suggestion, maybe even seasonal effects would have different seasons for
Sure.
months.
Sure.
Yeah, and sorry, last question. What are your next goal after knowing these discoveries that you have shared with us today?
our next goals are basically to explore this whole list of other opportunities we have here, right? I mean, I would, in working with the industry, we would like to see it developed as a drug and become available, so people don't use it off label. But I mean, you can buy this stuff now from Alibaba, and there are groups there on the web who are advertising it for brain doping, but that's really not a very smart thing to do, right? So it would be good to actually have the support of the clinical community and applying it to real problems.
Yeah. Sure. I think here, this side.
Hello, hello, Professor. Thank you for your nice talk. I'm Anna from KC in HKU. I want to know how you identify this E3B at the beginning, and is this E3B, this molecule, available in the herbal medicine or in the food, so that maybe we can take the food that include this E3B to for our cognitive improvement.
Yeah, well, hopefully once the clinical approval is granted, it will be used for conditions where you really need it, rather than just for learning before the next exam or some other recreational use. It came out of Carmela performed a screen using a cell reporter for the induction of the integrated stress response, and used that screen to screen through, in an academic setting, through 100,000 compounds, so not large scale. And this was the only compound in the end that carried through and became our lead candidate. And as I said, Calico and Denali have now improved upon it to make it more of a pharmaceutical than this true compound would afford us.
Okay, thank you.
Maybe, over that side,
Thank you very much for your very inspiring talk. My name is Mai from HKU. I have two questions. So first, have you considered any epigenetic analysis of this strip treatment? And have you considered any environmental factors of mice in different environments? And did you compare the mice treated from different ages? And if so, how does the mice, young mice and old mice, respond to this strip treatment?
Yeah, so
is there any difference?
Let me take the last part first, and that is, yes, we've looked at the old mice, right, and we improved their memories. We've also looked at the effect of the drug on, I mean, it makes you memorize things better, right? So that one of the issues where it becomes a problem, a potential problem, is in addiction, where if you remember the good things of taking a drug, right, the higher you're getting, better you may be more likely to become addicted, and that's indeed the case. So basically, if you do take ISRP in the future, don't take cocaine at the same time. Regarding the different environmental conditions, it's actually a very interesting point, because when I conclude here in my talk that we make a wild type, a normal animal smarter, right? They learn better, that we're improving upon evolution. This could be a very interesting point, or it could have a very boring explanation. And the boring explanation is that these mice are inbred, right? They have been raised and brought up in a deprived environment, so we may have bred them into stupidity, and then we're bringing them back into normal wild-type behavior, right? So whether evolution really hasn't optimized learning, I don't know, right, from this data. It's just in our experiment. That's always something we have to always take into account in our experimental system. They are experimental systems, right? And so it will be the future will tell us, right? How whether it is an improvement or not. Yeah. Mm-hmm.
Maybe over there. Last question.
Hi, my name is Jasmine from the University of Cambridge, Department of Obstetrics and Gynecology. This is really exciting work, very cool to hear about it. I was wondering whether you've considered administering the treatments in pregnant mice to determine potential teratogenicity, especially as pregnant women aren't historically included in clinical trials, and it'd be wonderful to be able to increase their inclusion.
Yeah. So we have not considered that so far at all. I think it's another very complicated issue. And I mean, right now the trial is in this rare disease, right, the vanishing white male disease, which is genetic. So basically the exchange factor I showed you has 10 different components to be built into a complex, and in that disease, both parents contribute a partially defective allele. And for some reason or another, these kids are born. They look healthy. And then once they experience a traumatic event like a fever or a mechanical injury, they fall out of bed or something, then the disease sets in that leads to severe degeneration of development, arrest of development. They lose their motor functions, and basically all the myelation in the brain goes away. So their brain melts. There's nothing you can do about them. So it's a very easy test, or a very, very easy clinical trial, because they would die otherwise within a few years. And even though it's a rare disease, it allows an easy pass into the clinic. And Denali wouldn't do it. So there's only a Calico Abbey trial right now. Denali wouldn't do it because there aren't enough patients and they couldn't make enough money, which I think is really, really, really sad.
Yeah, so before I end the session, I'm sure you all have further questions for Professor Walter and Professor Mori. Given so many young scientists and students here, can I ask you give an advice to the students? I'm sure there are scientific setbacks or challenges, hiccups when things don't work. What advice would you give them, from your research journey, your experience?
Well, my research experience is when things don't work and you don't know why they don't work, and you know you didn't screw up something that is obvious, things become exciting. And you should double down on that opportunity, right? Because once you have a paradox, something you cannot explain, and you figure out a way of resolving the paradox, then it cannot be boring, right? Because you couldn't explain it in the first place. So be adventurous. I mean, you all got into science because you wanted to explore more about how cells work, right? How nature works. So stick with that initial intuition. It's a fantastic job. Yes, it is frustrating. 95% of what we do doesn't work. But usually it's because we screw up one way or the other, or some instrument doesn't work, or right, your clone isn't the right thing, you have an extra mutation somewhere else. So it is an adventure, right? And where else can we be creative and we have so much fun when we go to the lab and be paid for it?
Mm. May I also ask Professor Mori for an advice to our young researchers here, what they could do to make an impact if they want to make an impact on modern molecular biology today?
I think first important thing is find a field which is very interesting to you, which fascinating you. Then, if you decide one field, then that means you are a newcomer. Otherwise, in other words, it's a latecomers. There are already many publications. And then I think my advice is find a good strategy that makes your own way. That's how I told this Eden story. We were latecomers. There are many publications, but I find a way for our team to go, that knockout and substrate. If we didn't take that strategy, we could not make any progress. So just find your own way. Read many literatures and then find a way, your own way. That's my advice.
Okay, thank you.
Let me add one more thing, and that is find a good mentor. Very, very important, right? And somebody who is not just selfish, using you as a slave to work harder, but rather somebody who has your career in mind.
Yeah, very useful.
It's all teamwork, right? Yeah. It's all teamwork.
Yeah, okay, good. So I'm sure you all felt like very rewarding this afternoon, hearing from Professor Mori and Professor Walter. Let's once again give a round of applause and thank you, the two very distinguished speakers today. Thank you.
Thank you, professors. Please be seated. It's definitely our great honor to have Professor Wong, Professor Mori, and Professor Walter with us today for the wonderful presentation. Time flies and we have come to the end of this session. We hope you have gained some knowledge and insights from our speakers. Now please your time to exchange further and network with all our speakers and also fellows. For our honorable participants and also our speakers who have enrolled for the next agenda, we will have the Victoria Harbour Tour and Welcome Dinner at West Kowloon Cultural District. We have arranged coaches to transport everyone to the Central Pier for the ferry boarding. So followed by the ferry ride, we will provide cold service to the dinner venue. Now please follow our event staff to the coach boarding area. For the laureates, please follow the staff to building at 10W. And for the young scientists, you may have your cold service at building 15W at 4:50 p.m. Once again, we would like to express our greatest gratitude to our speakers and moderator for your time. We are very happy to have every one of you in this session. Have a wonderful evening. Goodbye, everyone.